And some of you have already done the test, the quiz. Those of you who haven't, you have until midnight to get it in.
I had a question about the quiz.
Yeah.
It doesn't say anywhere on here. Is it like based on like last class section or like what, like where in the, when did you teach? Like what would this quiz would be on? Like,
Well,
just...
the first two subjects,
okay.
scientific inquiry and introduction to biology.
Okay.
I think that was sent to you as an announcement, what would be covered in this quiz.
Oh, okay. I was just wondering because it doesn't say on the quiz. Okay. Thank you.
Okay, yeah, make sure you always check your announcements because I send information like that out in announcements. Anything else?
I had another quick question. My name's Abigail Beck.
Okay.
Is there a quiz?
Welcome to Sick Notes. My name is
I can't hear you.
Is there a quiz weekly or is it just every once in a while?
Yeah, your microphone is cutting out.
I heard she said is the quiz weekly or is it like just every once in a while?
No, most every week there'll be something, either a quiz or a test, and there'll be 4 tests over the course of the semester. So the intervening weeks, most of the time, unless there's something that comes up, there will be a quiz.
Thank you.
Just out of curiosity, you might be curious about this. Remember we had that practice quiz?
Yes.
What was not graded, but I looked to see what the statistics were on it. Turns out the mean, the average grade for that was 57%. The high grade was 80%. The low grade was 20%. And the average time was 8.5 minutes out of 10 minutes that were allowed. So that was the practice quiz. You'll all do better on this one.
Professor, one thing, you had told me last, at the end of class, you were going to email me those 2 papers and unlock the quiz. And neither one of those things happened.
Well, I did it, but I didn't hear back from you that you didn't receive anything. So what may have happened is sending email in Canvas is a hit or miss proposition. And so if you didn't get them, I need to send them to your private email, because attachments often, not often, but sometimes attachments never make it.
Okay.
So if you still want them, I'll send them by regular email. If it's too late, I won't bother.
All right. I'll just send you an email from my personal email. That way you have it.
Yeah, please do that. And then I will send you there's 3 documents. I don't have quiz, that practice quiz is no longer in Canvas, so I'll have to send it to you as a Word document.
That's fine. I just wanted it to kind of see how I would do.
Yeah, send me that email right now, and as soon as the class is over, I'll send you those things so you can take a look at that before you take the quiz today. You have until midnight to take quiz 2.
The quick questions, Michael Soror. The quiz was out of 10 points. Will you adjust it in the grade book to 10 out of 10, or will you make it like not worth anything? Or
Today's quiz?
Quiz 1.
Quiz 1 wasn't graded at all.
Okay.
It's 0 out of 0.
Okay. I thought.
I think it shows your score, but it doesn't like actually add it to the grade.
Okay.
Yeah, it shows up in the grade book, but it's a 0.
Hi,
Out
my,
of 10 out of
oh.
on my grade book there. That's why I was just concerned, but okay.
Okay, someone else had a question.
yeah, hi, my name is Doris Garcia. So I just wanted to confirm that right now we should have like grade, it should be in Canvas, not available for this class.
The grade not available for this class.
Like basically we should have no grade at the moment.
That's right. Yeah. So if you
Okay.
look in grade book, you won't see any grade. The first graded assignment will be this quiz today, quiz 2.
Okay, thank you.
Professor, I did receive your email and it was buried in my spam filter.
Oh, okay. Did it have attachments?
Yep. It
So you
had
got
all-
everything.
Yeah, it had all 3 documents.
Okay, good. Then you don't have to
Sorry about
send me an email. Okay, so take a look at that before you take the quiz, especially the practice quiz. Okay, anything else? The main thing I want to do today is go over another slide set. This is on classification of organisms. And if I have time after that, I'd like to come back. I promised you we'd talk about life in the universe, and if we have time, I have a few slides on that topic. Do you see classification of organisms?
Yes.
Yes.
Should be gobbling up your whole screen. So how many different types of organisms do we have on this planet? A lot. Over 1.8 million species have been identified so far. And if you're curious about going over the catalog of them, here's a website for it. But it's important to keep in mind that probably at least that many, if not more, species remain to be identified. Many of them are microorganisms. And I'll talk a little bit later about how we identify microorganisms that won't grow in culture. So how do we keep track of all these millions, hundreds of thousands, and millions of different organisms? Well, we classify them. We divide them up into groups that show relatedness.
I'm not sure right now, so give me a message.
What's that? Is that a question? If it wasn't a question, please mute your mic. Okay, we'll have to talk about what does relatedness mean, by what criteria. And then we want to give each individual species an unambiguous name. And we'll talk about how that's done. Well, people have been trying to classify living things for a long time. Just to give you one example, Aristotle. He was a smart guy and he tried to organize things in a sensible manner. So he classified living things according to what he could see, the observable characteristics. And so he had animals and he had plants. Plants were fairly easy for him because he categorized them on the basis of size. Either big ones like trees, or middle-sized ones like shrubs, or little ones like herbs. So plants were nice and easy. Animals were more complicated. So his first criterion was, do they have blood or not? By whatever his definition of blood was. So those that he said had blood, were the mammals, those that gave birth to living offspring. So that was one category. Then he had those that fly, and among those that fly, he had birds. And then he had non-flying. So he classified them according to whether they laid eggs. So birds lay eggs and they fly. Other animals lay eggs and don't fly. And those are the amphibians and reptiles. And then he classified whales and fish. Among those who had no blood, according to him, were cephalopods like octopus and squid, crustaceans, crabs and lobsters. Insects were a separate category, but he made it too big. He included scorpions and spiders as insects, and they're really not. The shelled animals like snails and starfish, and then a category that he called zoophytes, which are animals that look like plants, like corals and sea anemones. So this type of classification was done not only by Aristotle, but many other people. And none of these schemes based on observable characteristics was satisfactory. Then along came Linnaeus. 1758, he published a book in which he proposed a binomial method of naming organisms. Binomial means two names. They are the genus and the species. This is the formal scientific name that every identified organism on Earth has, a binomial. And based on that, he proposed a hierarchical system of classification, which we still use. So what is a binomial for humans? Homo sapiens. This happens to be Christina Nettie Parker, the daughter of Catherine Parker, who's the chair of the biology department here. What's a binomial for your cat? Felis catus. Binomial for a killer whale? Or Orcinus orca. When using a name in written form, the genus, the first part of the binomial, is always capitalized, and the species is always lowercase. For example, the bacterium Escherichia coli capitalized the E on Escherichia, and coli is in lowercase letters. Both the genus and species are either italicized or underlined. So if you're working with digital letters, you italicize them. If you're, for example, writing something longhand, you can't italicize, so you would underline it as separate words. So in the case of the bacterium, Escherichia coli could be italicized, or it can be underlined, and the space between the words is not underlined. Now, once used in a paper, a scientific name is, after it's used the first time, after that, it's always abbreviated. And how is it abbreviated? E. coli. So you just use the first letter of the genus name and spell out the species name. So make sure you know this, you understand how organisms are named and how to write the names, because it will probably come up. In fact, I'm sure it will come up sometime on a test. Well, here's an example of how you do it. Streptococcus pyogenes is a bacterium. It's known to cause strep throat. The antibiotic amoxicillin can be used to kill most strains of S. pyogenes. So you see the point. The first time it's used, it's spelled out The second time, it's only the first letter of the genus and you spell out the species. Is it clear to everybody?
Yes.
Okay. Humans are natural classifiers. Think about your surroundings right now or what you see throughout a day. What are some of the things that you just automatically classify?
Grass.
Professor Patrick Dowell here.
Okay.
Kind of classify, like even just on the road driving, you're classifying like, oh, that's a truck, that's a car, that's a van, that's a big semi truck, that's a motorcycle.
Right, classify vehicles as you're driving down the road. And I'm looking what's behind you. It looks like you classify photographs.
Those are books.
You have books. Okay, that's a category. You classify some of the things in your office as books.
Yes.
Okay, what else?
Katrina the bird. You can classify like different subjects in school.
Like, yeah, sure.
Like,
Like biology and
yeah.
chemistry. Yeah, good.
Yeah.
That's classification. What else? Someone else speak up.
This is Bennett Taylor, and I was saying plants and grass and trees, stuff that you see like in your backyard.
Okay, you could say everything that's green gets classified in a category.
Okay.
What else can you think of?
You could do like,
Oh, you can go.
I was gonna say you could classify like dogs, cats, and animals in
Right.
general.
Okay, good. So we've classified plants and we've classified animals. What else?
My name's Kaylin Hilberg, and we classified colors.
Classify colors. Yeah. I mean, when you say colors, so what are you thinking of?
Like how there's different shades of orange, but we still say that it's orange.
Oh, okay. Yeah. Yeah, that's good. What else can you think?
Professor, my name is Zara.
Zara Nazari, okay.
Yes, I think by the sizes too.
Classified by size, yeah. That's what Aristotle did when he got to plants. He had big ones, little ones, and medium-sized ones.
Professor, my name is Samira. Classified of like a food, vegetables, fruits, green leaves to eat.
Every time we eat, we classify the foods.
Yes.
Good.
Hi, my name is Doris Garcia. I was gonna say we classify music, like music genres.
Yeah, that's very important. You separate out music types. Yeah, well, you go ahead.
Okay, I'm not sure it is correct, but I think is that it's also classified like a brain, like you know, like the front or back of the brain, like hold the information and stuff like that.
Different portions of the brain are classified as different, having different types of functions. Yeah, yeah, that's a type of classification. Yeah, so we're getting the idea that as we go through a day all around us, we automatically classify things. And certainly that's true about all living things. We'd like to classify them all. And so how do we do that? Aristotle and people coming after him who just classified what they saw ran into problems. It became too cumbersome and there were too many inconsistencies. And along came molecular biology and the ability to sequence nucleic acids. So now we have very precise ways of doing it based on ribosomal RNA. And I will show you that in a little bit. Using ribosomal RNA, all living things are classified into one of three domains, either bacteria or archaea or eukarya, the eukaryotes. And the eukarya are divided into plants, animals, fungi, and protists. Protists is kind of a nebulous term, and it really comes down to any eukaryote that doesn't fall into any other category gets classified as a protist. Now, bacteria and the two major domains here to the left, what's the difference between them? First of all, what are the similarities? Well, they're all prokaryotes. They're all single-celled. They all reproduce by binary fission, which means just splitting in two. The cell just splits from one into two cells. They all have ribosomes. They all have cell walls. And they all have their DNA loose in the cytoplasm in a nucleoid. So as for representing two different domains, they have a lot of similarities. So what's the difference? Well, they have different structures in their cell walls, the pseudopeptidoglycan wall and the archaea, and a lipopolysaccharide is the main component in the walls in bacteria. The main distinguishing feature for me between these two is their habitat. Archaea live in extreme environments like hot springs and salt lakes, harsh environments, whereas bacteria by and large live in temperate environments. Archaea do not make spores. That is, they don't have a dormant resting body, whereas bacteria do produce spores that can live for long periods of time without metabolizing. And when they have the right conditions, they can germinate and grow. So what you do when you classify is to take things and put them in boxes and then keep dividing those boxes into smaller and smaller boxes. So we have our three domains here. And we have a box, and we're going to say, we're going to put all the eukaryotes in that box. So we have a kingdom that we call protists, another kingdom we call fungi, another one we call plants, and one that we call animals. Then if we divide, if we take one of those and say just the kingdom Animalia, the animals, and we start dividing it, and we sort that out into groups called phyla, one phylum, multiple phyla, and there are 8 of them, 8 different categories of animals. And so if you list all the different categories for classification, you have the list on the left here, starting with the domain, and that's divided into kingdoms and phyla, class, order, family, genus, and species. And the unique name for every organism is the binomial genus and species. So let's go through an example. We have 3 species here. Number 1, number 2, and number 3. If we look at the domain in which they are classified, they're all eukaryotes. They all have true nuclei. If you look at the kingdom that they fall in, they're all animals. And if you look at the phylum that the kingdom is divided into, they're all arthropods. But at the level of class, the first one is a crustacean. The other 2 are insects. The level of order, the first is Decapita. The other 2 are Diptera. And finally, when we get down to the level of the family, each is discrete. Caridia, Drosophila, and Nematocera. The genus and species, the first one is Homerus americanus, the second one is Drosophila melanogaster, and the third is Aedes aegypti. Okay, what are these animals really? Well, Homerus americanus is a crab. Drosophila melanogaster is the fruit fly, and Aedes aegypti is a mosquito. So each has a unique name. Each is an individual species. Now, I have to expose you to my sense of humor on the next slide. We'll see if anybody laughs. So fruit flies are a very popular model organism for geneticists. Many geneticists work on fruit flies. And if you go around and visit Drosophila labs in various places, almost invariably on the wall somewhere, you'll see a sign that says something like this: time flies like an arrow, fruit flies like a banana. Now, what does it mean? Doesn't mean anything. It's silly, but it appeals to the sense of humor of certain types of geneticists. And then there's always somebody that messes it up, like on the right here. Time flies like an arrow, fruit flies like a banana, house flies don't like going flat. That means if you swat them, they go flat. Okay, that was my attempt at amusing you. Whether you were amused or not, I'll go on. So what criteria do we use to classify? I've already said that we use RNA species and RNA sequences. And these are used to establish phylogenies. So here we have on the right 3 species that all fly: a bird, a bat, and an insect. How do we classify those sorts of things? Well, structurally, a bird and a bat are similar. They both have wings attached to their legs. Whereas if you look to the right here, an insect does not have bones in its wings at all. But if you look at this sort of primitive phylogenetic tree here, somewhere back in evolution, an organism evolved that had 4 limbs. And it split to 4 limbs evolved into wings in the bat and the bird, but mice and crocodiles also have 4 limbs, but they don't have wings. And so structurally, the wings in the bat and the bird are similar, even though the bat and the bird are not closely related phylogenetically. A bat is more closely related to a mouse, and a bird is more closely related to a crocodile. So that brings us to the terms analogous and homologous. What do they mean? Analogous means similar function, but not evolutionarily similar. So bird wings and insect wings have a similar function. They don't have a similar origin. Homologous, on the other hand, is the opposite. Homologous organs or structures have a similar evolutionary history, but don't necessarily have a similar function. So I've written it out in more detail here at the bottom. Bat and bird wings are homologous structures, indicating that bats and birds share a common evolutionary past. In contrast, analogous structures like the wing of a honeybee is similar in shape to a bird wing or a bat wing, and it serves the same function. However, a honeybee wing is not composed of bones and has a distinctly different structure and embryonic origin. So these wing types illustrate an analogy. They're similar structures that do not share an evolutionary history, and similar structures with similar functions. Okay. Do you see the sharp distinction between an analogous structure and a homologous one?
Yes.
Yes.
Okay. I won't belabor the point. On the contemporary bases for classification, morphology, it does not have much to do with it. As I've just pointed out, ribosomal RNA has a lot to do with contemporary classification because of the molecular precision. So why is ribosomal RNA so useful for classification? First of all, it's found in all organisms. Prokaryotes, eukaryotes, bacteria, animals, plants, whales, trees, everything has ribosomal RNA. And within ribosomal RNA, some of the regions are highly conserved, some are highly variable. You can see that here is a ribosomal RNA molecule with the sequence of bases indicated. The red residues are highly conserved, whereas the blue and green ones are highly variable. So if you want to distinguish between two closely related organisms, you'd look at these highly variable sequences to look for differences. Whereas if you're trying to classify on a gross level, you would look at the highly conserved, or the red bases. And you can see that here on the right, where there are three ribosomal RNA sequences from mammals, human, mouse, and rat. And you can see that they're very similar, if not identical, to each other. Whereas if you look at the same sequences from bacteria, you can see that there are a lot of differences, even though there are some highly conserved regions. All the yellow ones are highly conserved. And these are probably bases that are indicative of living things. All living things have the yellow, and if you start discriminating among species, you have the greens and the reds. The highly conserved sequences indicate important functions across species. So, sequencing ribosomal RNA gives us a phylogenetic tree representing the bacteria, the archaea, and the eukarya. Now, the bacteria, most of them are single-celled and do not make their own food. Some of them do, like the cyanobacteria have chlorophyll. The green filamentous bacteria have chlorophyll, so these are food producers, all the rest are consumers. The archaea are extremophiles. For example, the halophiles like salt, they'll grow in the salty regions. The methane-loving bacteria either can grow on methane or can produce it. And then usually a bacterial name that starts with thermo means that it grows in a hot place. Reminds me, I once worked for a company that gathered environmental samples from all over the world from extreme environments. And one of the samples was from a hot spring. It was a bacterium that would only grow in boiling water. If the water was any cooler than boiling, it stopped growing. So it's an example of an adaptation to an extreme environment. Domain bacteria, here on the right, you see the three basic forms that bacteria have, the spirochetes, the rods, and the spheres, the cocci, the bacilli, and the spirochetes. Bacteria are found everywhere on Earth, including the polar ice caps. Most are environmental organisms. Some of them are pathogens. So for example, there is a spirochete, spiral bacterium that causes syphilis in humans. Domain archaea, I've gone over these and compared archaea to bacteria in an earlier slide. Just to review, they're all prokaryotes and single-celled. They're extremophiles, live in very strange habitats. They are now known to be ubiquitous on Earth. They are everywhere. Most of them are not culturable. So you can take a sample from somewhere that has an archaea in it and try to grow it in a petri dish and you'll get nothing. So how do we know they exist? Well, they're known from metagenomics. Does anybody know what metagenomics is? Don't expect you to, but somebody might have heard of it. Well, metagenomics is based on isolation of nucleic acids from raw samples from nature. So you might sample soil, you might sample a cow's stomach or a termite gut, but it's just a raw sample that you take the nucleic acids out and sequence them. And then start stitching homologous sequences together. And after a while, you do this enough times with enough computers, you'll put together the entire genome of various different organisms. And based on that, you have a genomic sequence, you can predict what the organism is. So many of the archaea are known as a result of metagenomics. None of them is known to be pathogenic to anything, although they themselves can be attacked by viruses. And here at the bottom, there are pictures of environments where you find, for example, thermophiles in a hot spring, acidophiles in an acid lake bed, and halophiles in a salt lake. Archaeal cells come in a lot of different kinds of shapes. Some of them are strange. And then we come to domain eukaryotes. Animal cells, plant cells, they range in size from 10 to 100 micrometers, divided into the 4 kingdoms that we've talked about before. The right, 2 pictures. What are they? Anybody recognize either of those pictures from what we've said before in the class?
One. Go ahead.
Aren't they like cells? Like
They're cells, yes.
One is
Yeah,
muscle
but-
tissue, the bottom picture.
Bottom picture is what?
Muscle tissue.
Bottom picture is muscle.
I would say that's a plant cell.
Oh. Well.
The top one might be a muscle.
Okay. Is that Patrick said that?
Yes, sir.
Justin said that.
Yeah, so you think the top is an animal cell, and why do you think that?
I think the top one's an animal cell, and this is Justin Hansen because they're not organized, and then the bottom one's organized, so that's a plant cell.
And what accounts for the difference in shape?
The plant cell has the cell walls, or so they're like more organized. I don't know.
Right, you're right.
And they're
Cell
rigid.
wall makes the cell rigid and orderly, whereas animal cells don't have walls and they can take a lot of different kinds of shapes. Protista, as I said, is kind of a grab bag of anything that doesn't fit anywhere else. It's called a protist, like amoebae and volvox found in the ocean. More examples of protists, kelp, algae, oomycetes. Those of you who may have had biology labs may have looked at paramecium and amoebae under microscopes, and trypanosomes, which are parasites. Fungi. Fungi are unusual in that they are the degraders of the planet. If it wasn't for fungi, the planet would fill up with detritus. Fungi are able to degrade virtually any biological material. And how do they do it? Well, they have very powerful digestive systems, but not inside their bodies. In essence, their stomachs are outside of their cells. And by saying that, what I mean is that they produce digestive enzymes, but then secrete them into the environment. And if the enzymes find substrates in the environment that they can digest, then the fungus will take up the digestion products and use them as food. So that's how they degrade and benefit from degrading organic material. This particular fungus happens to be penicillium, which produces the antibiotic penicillin. So what you see here is a body cell, a hypha or a mycelium, and these little blue balls are the spores. The spores fly around. Penicillium spores are everywhere, including in your refrigerator. This probably isn't penicillium, it may be aspergillus, which is a close relative, but this is a good example, a good illustration of how fungi degrade things. So this is probably an orange, used to be an orange, and it was fine. Penicillium and aspergillus spores are everywhere. They're all over you right now. They're all over your food. And you only notice them when you give them an opportunity to grow. So as this orange aged, began to leak nutrients, then aspergillus started growing on its surface, and finally it was a full-blown mold and ruined the fruit. There are many types of specializations among fungi, and this picture on the lower left shows one specialization. This is a fungus that grows in the soil, and it's known as a nematode trapper. And what you can see here is that it has trapped a nematode. Nematodes are little worms, usually microscopic, that live in the soil. So what this fungus does is make these traps, 3 cells in a ring. And if the nematode comes swimming along and happens to enter that ring, it snaps shut. These cells then start making digestive enzymes, secrete them. They digest the nematode and eat it. So it's an example of a sort of an extreme adaptation for getting food. How else do we interact with fungi? They're very much involved in food production. So the yeasts are employed for making beer, wine, and bread. Some of the filamentous fungi make cheese. So for example, Penicillium camemberti makes camembert cheese, which you see here. P. roquefortii also is used for cheese making. What do? They impart flavor to the cheese.
Professor Yoder.
Yeah.
This is Bennett Taylor. So is this the same classification of fungi that Alexander Fleming used to come up with penicillin?
Yes, it's the same genus and same genus, but a different species. So yeah, Alexander Fleming, because Penicillium flies around and is everywhere, he was annoyed that it contaminated his bacterial plates. Excuse me, I need to get a drink of water.
Estas bacterias no son.
So Alexander Fleming was perceptive enough or curious enough to notice that this penicillium that was contaminating his plate had no bacteria near it.
No son nocivas para
And from that, he concluded that the penicillium must be making something diffusible as inhibiting the bacteria. And he was exactly right. That diffusible substance was penicillin.
So I got a question. These bacteria are not harmful for human health?
No, most.
Because they're in the food, no?
Yeah, fungi are everywhere, and if you eat them, they don't harm you. Most fungi cause no harm, but some do. Some produce substances known as mycotoxins, which are extremely toxic. Some of the most potent carcinogens known are produced by fungi. So the point is, don't eat a fungus unless you know that it's safe. So if it's in your cheese, it's safe. You see in the lower right here, mushrooms. Some of them are delicacies, and others are deadly poisonous. So never eat a mushroom unless you know that it's safe. If you see mold growing on your bread, you can cut the moldy part off and still eat the bread, but don't eat the moldy part.
Professor, I want to say something. So you say about the mold. So what about the, do you know like a dry persimmon? They have also like have a mold, and I think people can eat that, like a dry persimmon, a fruit.
Persimmons?
Yeah, a dry persimmon.
I don't know about that, but I know an example like that, which is so-called royal wines, where fungi are allowed to colonize. In this case, it's a Botrytis species, allowed to colonize the grape berries. And the grape berries then are crushed up along with the fungus. And what the fungus does is causes very high concentration of sugar. So if you've ever had a dessert wine, often called a royal wine, it's because the fungus has concentrated the sugar in the wine. And in that case, the fungus is completely harmless.
Professor?
Yeah.
I have a question, Leanne Metzger. So how does allergies work into it? Like, why is it okay for some and then allergies play in factor for others?
Why are some people allergic to fungi?
Yes. Like penicillin or things like that, or gluten allergies or.
Yeah, this is really an area of immunology. If your immune system reacts to an antigen like, say, a fungal spore, you can have a cytokine cascade and be extremely allergic to that. And this is a function of individual people. Most people do not have allergies of that type, whereas some people do. It's not a very good answer to your question, but that's all I know about it. Anything else? Fungi, by and large, are our friends, but we have to be careful because they sometimes can be very dangerous. Okay, we get to plants. Plants are the great producers because they have chlorophyll. Chlorophyll is the only pigment that can capture sunlight and convert it into chemicals. And we'll talk a lot more about photosynthesis later in the course. Animals exist only because plants do. Animals cannot produce food. All they can do is eat plants. And as you see here, animal kingdom is divided into 8 different phyla. And the phylum Chordata down here at the bottom, Chordata is us. It's all the animals with backbones. Okay. That's the end of this slide set, and I'm almost out of time. When you have a quiz, I subtract the quiz time from the lecture time so that I'm not taking extra time out of your day. I've only got about 7 more minutes, but what I'd like to do is briefly go over another set of slides which I find interesting that have to do with life in the universe. Do you see the universe?
Yes.
Yeah.
Okay. All right. Since we don't have a lot of time, I'll try to go through this fairly quickly, but I have a question, and I suspect you do too. You wonder, we have all these telescopes and we can see the magnitude of the universe and how many planets and galaxies and such there are out there. Is there life elsewhere beyond Earth? So let's just consider that for the next few minutes. And just to remind you the scale of things, this is the scale of life forms on Earth. And then if we want to go outside beyond the planet Earth, we use telescopes like this. And remember this image, this space contains 15,000 galaxies, so 15,000 Milky Ways, for example. And one galaxy can contain billions of stars in one galaxy. So the number of stars and planets is almost incomprehensible. Now, most stars are likely to have planets. And what is the probability of life on them and what conditions are required for life? There is a subdiscipline known as exobiology. Exobiologists look for life elsewhere in the universe. And 2 of the things that they assume are necessary, liquid water and an atmosphere that should contain nitrogen and carbon dioxide. This is a picture of the space telescope Kepler, which was launched in 2014 and retired in 2018. Its goal was to go as far out away from our solar system as possible and look for planets in other solar systems and other galaxies. So water must be liquid. And here you have a thermometer showing where water boils and where water freezes. So the assumption is that any planet has to have a surface temperature somewhere between 32 degrees and 212 degrees Fahrenheit. The Earth does. Most of the planets that Kepler found do not fall into that category at all. So temperature is one of the critical factors. And temperature is determined by the size of the star, the sun, and the distance the planet is from it. So there's what's called the Goldilocks conditions for life. Remember, Goldilocks can't be too hot or too cold, has to be just right. The same is true for planets. They can't be too hot or too big, or too cold or too small. They have to be just right. So Kepler, well, first let me go through what's on the left here. Temperature has to be, allow for liquid water. There should be an atmosphere. There should be a liquid core to the planet, which causes a magnetic field. And this is important for life because it deflects radiation. So there'd be no life on this planet if we didn't have a liquid core to the Earth, because the sun's radiation would destroy all living things. And the mass is critical. If it's too big, the gravitational force is enormous and it would crush living things. If it's too small, it can't hold the atmosphere. There's not enough gravitational force to hold the atmosphere, which is critical for life. So Kepler found a planet called Kepler 186f. And you see the solar system for Kepler 186 here. F is way out on the edge. There are other planets in closer to the star. And compared to our solar system, where we have a sun which is bigger than Kepler's sun, so it gives off more heat. And we have Mercury, Venus, and Earth, and then Mars is out farther than that. So we know that life exists on our Earth. Question is, does life exist on Kepler 186f? So there are two problems in trying to determine if life exists. One is distance. So this slide shows you the distance between various objects that we're familiar with. You remember that the speed of light is 186,000 miles a second. So the distance to the moon is 1.5 seconds. In other words, it takes 1.5 seconds for light to get from the moon back to Earth. To the sun, it's about 8 light minutes. Earth to Mars is 12.5 light minutes. Earth to the nearest star is 4.3 light years. So this is an enormous distance just to get to the nearest star. And to get to the other side of our own Milky Way, remember planet Earth is way out here on the edge of the Milky Way. It's 52,000 light years across just our galaxy. So these are distances that we just can't imagine. They're so great. Kepler 186f is 500 light years from the Earth. So that's 2.5 times 10 to the 15th miles. It is in the Milky Way, but the time to get there from Earth, if you could travel at the speed of light, it would take 500 years to get from Earth to Kepler 186f. But we can't travel at the speed of light. We have rockets that go much slower than that. The fastest of which, to escape the sun's gravity, would take 242,000 years to get from Earth to Kepler 186f. So how are we going to find out if there's life on Kepler 186f, just because it seems like it has the conditions in its solar system for life to start, life to form? So distance is a huge problem. The other problem is, what is the form of life that we might expect to find on another planet? I've gone through some calculations here that I won't take time to detail now, just to say that in the prebiotic ocean, life formed on this planet. And the forms of life that existed on this planet through most of its existence are very primitive, like trilobites, thermal bacteria, thermal vent bacteria. Highly complex forms of life have only been in the last few hundreds of thousands of years, or about 0.0001% of the history of the Earth itself. So the conclusion is that any life forms on other planets are likely to be non-intelligent, likely to be very primitive. You can see that displayed here on this time scale, which is in billions of years. The Earth is about 4.5 billion years old. At about 3.5 billion years, life forms started to appear. 2.1 billion years, more complex life forms, multicellular ones, appeared. About 1 billion years ago, much more complex life forms appeared. This is all in the primordial ocean. 600 million years ago, we got really complex vertebrates in the ocean. 360 million years ago, we got vertebrates on land. And only about 7 years ago, we got 7 million intelligent beings, the primates. Humans are only 0.4 million years. So that's just really right on top of the zero here. It's almost very little of the history of this planet has had intelligent life on it. So now if you say life on other planets has similar distributions of types of life, most of which, through 99.999% of the planet's existence, is unintelligible, then that life is not going to come looking for us. And we're very unlikely to find it, because you have to actually go to the planet to dig it up. Incidentally, I heard just today that the US has a landing on Mars today. It's going to scoop up samples and look for evidence of life on Mars. Anyway, the likelihood of life, living things, is much higher than that for intelligent life. So I'm keeping you over time. These are not really conclusions. They're really my opinions about things I've read. Is there life elsewhere? The probability of that seems to me to be very high. And the reason I say it's very high is because the number of Goldilocks planets in the universe is probably astronomical, just because there are so many planets, so many stars, that it's very likely that conditions for formation of life are common throughout the universe. However, so the probability of life is high, likelihood of detection by us, low. And why is that? Well, because the distances are too great. To get to the nearest star is light years away. And even if we could travel the distance, what we'd very likely find is a very primitive form of life that we couldn't really communicate with. So the question then is, will we ever detect extraterrestrial life before we ourselves become extinct? It's a question you can think about. We know that we will become extinct. Don't know when, but given the climate change that we're dealing with now, it's something to worry about. But no matter what the time frame is, if our species is going to, what's that? Don't know where this came from or how to get rid of it.
I think someone share the screen.
What?
Somebody's screen sharing their screen.
Oh, could you stop screen sharing, please? Anybody know how to get rid of this?
Someone just started sharing their screen. There you go.
Okay. All right. Well, we're going to have to, if we're going to survive over the long term, we're going to have to leave this planet because the planet itself is doomed at some point. So think about space stations where we could travel through space and exist beyond our planet. But that's all fantasy, and I've gone way over time, so I apologize for that. Are there any questions before I end?
Sorry. No, I just had a comment. I was gonna say, I think there's a way for you to go in and be able to make it so that nobody could share their screen, and also so that everyone is muted when we start class as well, until you allow them to unmute themselves.
Okay, I'll go into my settings. I think I remember seeing that about screen sharing. And I'll make that change. And also the mute. Okay, don't forget, take the quiz before midnight. Anything else?
I had one question on the first quiz. One of the questions that was on it was talking about the smallest, most fundamental unit of matter. And you had